Good morning, children, and welcome to the online teaching session. Students, in the previous video, we were discussing about the structure of DNA. In that, we saw the detailed structure of DNA and some points that was given by the Watson and Crick. Now, two more points are left in this case, and these are called as the rules of the DNA. Number one rule is base pair rule. Okay. Now see, children, what are the bases in the DNA? We have already studied that DNA is made up of pentose sugar, then phosphoric acid and nitrogen bases. So, we have two bases in the DNA, and the two bases of nitrogen are purines and pyrimidine. Okay, as I have already told you that we have the two purines, and the two purines are adenine and guanine, and the two pyrimidines are cytosine and thymine. Okay, so see what happens in case of base pair rule. In case of base pair rule, it is written that purines always pair with pyrimidines. This we know very well. Even in class 10th also you must have read this thing. Okay. But here we have to see, or it comes under a rule that is called as a base pair rule. Now what is the rule? Purines always pair with pyrimidines. And in case of purines always pair with pyrimidines means A always pair with thymine and cytosine always pair with guanine. Okay. Now see, this always pairs with this with double bond, and cytosine always pairs with guanine with triple bond. Clear? It is paired with triple bond, and this is the reason that the DNA strands are complementary to each other. So because of this, what is the conclusion? DNA strands are complementary to each other. Okay. So if the question of 1 mark comes that give reason why the DNA strands are complementary to each other, then we can write down, because it is according to the base pair rule, purines always paired with pyrimidine, where A pairs with T, C pairs with G with triple bond, and this with double bond. Okay. So this is about the base pair rule. Now, your next rule is Chargaff's rule. See, the next is Chargaff's rule. Now what the Chargaff has said us? Simply, Chargaff told us that A and T is equal, okay, C and G is equal. Now I can write it like this. A is equal to T and C is equal to G. Now here don't get confused, children, that I have drawn bonds. This is not bond. This is the symbol of equal. Okay. So A is equal to T and C is equal to G. Clear? Now suppose, how we can understand this? Suppose these are the two strands of DNA. If it is 5 prime, this will be 3rd prime. This will be 5 prime and 3rd prime. Now see, A pair with T, C pair with G. Okay, once again I am writing T pair with A, C with G, G with C. Suppose if I have drawn like this, then how many adenines can you see? 1 and 2. Then how many thymines are there? 1 and 2. So what we can write down in this case? 2 is equal to 2 in this case. Okay. Now you come to this portion. How many cytosines are visible to you? 1, 2 and 3. So how many cytosine? 3 is equal to. Now this is the symbol of equal to. This is not the triple bond because triple bond I have already made here. Then how many guanine will be there? 1, 2 and 3. So 3 is equal to 3. So what the Chargaff has said us that A will always be equal to the T and C will always be equal to the G. So after doing all this, he got a conclusion that A by T is what they said. They said that A by T is equal to 1 and C by G is equal to 1. So this is called as the Chargaff's rule, that number of, now they said that base pair, this will always pair with this will always pair with this. But Chargaff said that the number of purines is always equal to the number of pyrimidines. Now in this case, one question is very common, either in your medical exams or in your board exams of 1 mark. Now see, they used to give you any one of these in the form of ratio, and then the examiner asked the ratio of the other, the percentage of the other. See how. Okay, now the rule is okay, that is A by T will be 1 and C by G will be 1. This means that A is equal to T and C is equal to G. Now see what type of question comes in the exam generally. Okay, and the question contains 4 mark or some marks in case of medical, but the answer is very easy. Suppose in the question if it is asked that the A is equal to 20%. Okay, the percentage of adenine in the structure of DNA is 20%. Now, what is the percentage of anything we can write down? Guanine. Okay, then how we can find out? Definitely we will go to the base pair rule as well as the Chargaff's rule. According to the base pair, what happens? A always pairs with T. And according to the Chargaff rule, what happens? A and T is always equal to 1. So, if A is equal to 20%, then T will also be equal to 20%. Okay. Now what is left, children? The left is C is equal to G, or we can say that C by G is equal to 1. Now, out of 100, 40% is already gone. How much percentage is left? 60%. Clear? So, according to the Chargaff rule, C is equal to G and their ratio is same. If their ratio is same, it means C will be 30% and G will also be 30%. So, our question was, what is the percentage of guanine in the structure of the DNA? We'll write down the percentage of the guanine is 30%. Clear? So, this is all about the base pair rule as well as the Chargaff's rule. Now, children, our next topic will be structure of RNA. Before we start the structure of RNA, we should understand the meaning of the difference between the RNA as well as the DNA. I think so. I have taught you this thing earlier also, but here we will see this thing in detail. What is the difference between the structure of DNA and RNA? Okay, what we can write down in point number 1 about RNA. We can write down this DNA is the genetic material, but it is not the genetic material. Now, why it is the genetic material? Because it has one functional group. This is the important part. One functional group at carbon number 3. Okay? And it has two functional group at carbon number 2 and carbon number 3. Okay? And this is the reason that it is the genetic material and this is not the genetic material. Now what happens in this case? This is highly stable. If it is genetic material, it should be highly stable. And this is not stable in comparison to the DNA. Clear? Now, in this case, A is always paired with T. And in case of RNA, in pyrimidine, T is replaced by uracil. So what we can write in this, T is replaced by uracil. Okay, so A will be paired with U. Clear? If there is T, that will be paired with A will pair with T, but here A will be paired with U. So all these are some important differences between the structure of DNA and structure of RNA. Now, we have to start the structure of RNA and its types. This we will do in the next class. Thank you, children.